Hello, my dear children. Welcome back with another video. Okay, it is of ICSE class 10th biology video. So it's been a long time ago. I said that I made a video on this particular topic, but sorry for that. I'm apologizing for it. I can't make because I went to again I went to somewhere. Okay, since this is the vacation time, so I'm just roaming. Okay, so yeah, now it's time to look at that particular topic of ICSE class 10th biology. Okay, so today we are going to deal with ICSE class 10th biology chapter structure of chromosomes, cell cycle and cell division. Okay, so today we are going to deal with this particular topic. Yep, children, this is the second chapter and the first chapter is the cell. Okay, cell. So this is that chapter, that first chapter. It is just for revision only. Okay, so there's no need to study more thoroughly. Okay, so just I'm telling you that what is cell. You all know cell is nothing but the structural and functional unit of life. Okay, all the living cells are made up of cells, isn't it? And even our own life start with the pre-existing cells that are known as zygote. Okay, so I hope these are all the things. Okay, so yeah, let's move to this but main topic. This means this is the main chapter and this is the new chapter as well. Means you are dealing with this chapter for a very first time. Okay, structure of chromosomes, cell cycle and cell division. Okay, so yeah, without wasting no time, let's get started with these topics and let's understood one by one all the topics very nicely and very deeply as well. So yeah, if you as usual, if you love this video, so please like my video, and if you didn't subscribe the channel, so go and subscribe it first and press the bell icon so that you get every notification after posting my every video. Okay, so yeah, let's see what are structure of chromosomes, what are cell cycle and cell division. Okay, what is this chromosome? Let's see that first. Okay, so what are chromosomes? But see, when a normal unstained living cell is observed under light microscope, its nucleus may not appear to contain any particular thing inside. Okay, let me explain you in Hindi. Okay, according to your convenient. जब भी हम सेल को देखते हैं माइक्रोस्कोप में, जब भी हम सेल को देखते हैं, सो जो सेल होता है उसका न्यूक्लियस हमें नहीं दिखता है, इजंट इट? उसका न्यूक्लियस हमें नहीं दिखता है. क्यों नहीं दिखता है? क्योंकि उस पर अभी तक कुछ स्टेन नहीं होता है. स्टेन का मीनिंग क्या होता है? कोई डाई उस पर नहीं लगी होती, कोई कलर नहीं लगा होता है. ओके, जब हम अब सपोज आपने ऐसी किसी भी पार्ट को रख दिया. ओके, लेट टेक अनियन पील. ओके, आई होप आपने ये वाला एक्सपेरिमेंट किया भी होगा. You had done this experiment as well. So let's take an onion peel, isn't it? So if you keep this that onion peel in the microscope, okay, so you might not seen anything. Okay, but when you stain it, when you color it, okay, when you color the onion peel and then you keep inside the microscope, you can see each and every part very thoroughly, isn't it? You can see each and every part very thoroughly. Why? Because unstained things cannot be observed by the light microscope. Okay, its nucleus may not appear by because they are unstained, they are not colored. Okay, but when you stain with some dye or with some color, you can see that particular thing inside the nucleus. Okay, the function, the structure and the minute structure even you can see inside the microscope, isn't it? So for visualizing the things inside the microscope, you need to stain the thing. Okay, you need to apply some dye or some color on this. Okay, so this is the thing first we are going to start with. Okay, now the nucleus contains most of the cell's DNA, which is organized into discrete units called chromosomes. Now, what is chromosomes? Here, I'm telling you in a very good manner. Okay, so chromosomes are nothing but they are the condensed form of chromatin fibers. Okay, they are the condensed form of chromatin fiber. Now, what is this chromatin fiber? So that we'll see right now. Wait. Okay, just wait. Okay, so each chromosomes contain one long DNA. Okay, dinucleic ribonic acid. Okay, DNA full form. So DNA molecules associated with many proteins. We have many proteins on DNA. Okay, this complex of DNA and protein is called the chromatin. Okay, means when DNA and protein both mix, okay, both they mix, they form the chromatin. Okay, and when a cell is not dividing, the chromatin under an electron microscope appears to be in a form of very long and extremely thin, darkly stained fiber. Hence, what is the meaning of chromosomes? Chroma means the color, and the soma means body. Means the colored body are refers to chromosomes. Or if you want to write the chromosomes in your examination, so you can take you can write the chromosomes nothing but the condensed form of chromatin fiber. They are the coiled and condensed form of chromatin fiber. Okay, firstly they were in a chromatin fiber, in a form of chromatin fiber, then they become chromosomes. Now, where does this chromosome present? They are present inside the nucleus of the cell. You have cell, then nucleus. Okay, let me tell you. Suppose this is your cell. Just wait, children. Okay, suppose this is your cell. Okay, this is your nucleus. This one, this is your cell. Okay, this is your nucleus. Okay, and inside this nucleus you have, let it draw here. Okay, you have this type of structure. You have this type of structure. Okay, not this type of structure, even you have this type of structure, sorry, this type of structure. Okay, very in a very coiled form, in a very much condensed form, isn't it? Like this you have, this is your what? Chromatin fiber. This is nothing but the chromatin fiber only. Okay, when the chromatin fibers become condensed, means become thick. Okay, become thick and short. When these chromatin fiber become short and thick, like this. Okay, so they will rapidly change into chromosomes. Okay, they will rapidly change into chromosome. This is not F. Okay, this is your chromosome. Okay, like this it is present in a pair. Okay, like this it is present in a pair. Okay, now what is chromosomes? As I told you, chroma means, let me write here, chroma means color, and soma means body, means the colored body refers to the chromosomes. I hope this is clear to you. Okay, so what are chromosome? Chromosome nothing but the condensed form of the chromatin fibers. Okay, chromatin fibers are the thread-like structure, this very thread-like structure. Okay, they are very thin. Okay, so sorry, they are very thick, no, thin, sorry. So yeah, let's see the discovery of chromosomes. I hope chromosomes are clear to you. Okay, now what is the discovery, means how chromosomes discover, how they come to the existence. Okay, so chromosomes was firstly discovered in 1882 by a German scientist known as Walther Fleming. Okay, the name of the scientist was Walther Fleming. Very crucial. Okay, mark it down. Write the name in your text, that Walther Fleming was the scientist who discovered this chromosomes in 1882. What he means, how they discover, how they know that this is the structure of chromosomes. So what happened? First, they means before when they was doing some experiment or some research, so he saw he saw in a microscope that the cell of the larva of a salamander is a reptile. It is a reptile. Okay, so no, it is not a reptile, it is an organism. Okay, salamander. So what happened, he saw the larva of salamander, which is an organism. Okay, so that organisms, a larva, the salamander larva, the larva or the cells of the larva are rapidly dividing, they are dividing very fastly. Okay, he noticed this. Okay, that the larva of this salamander's the cell of the larva of salamander are rapidly dividing. Okay, so and he saw a minute thread like appear to the dividing lengthwise flaming called their division mitosis. So what happened, he saw that the dividing cell are forming in lengthwise, they are means they are dividing in a lengthwise form. Okay, so he called this division mitosis. Okay, who Fleming called this division mitosis, which actually means the thread. Okay, mitosis means the thread. Now subsequently, with the help of more powerful microscopes, because since at that time the microscope was very old and very means it was not in advanced way. Okay, that time's microscope, that's why he can't see anything in a clear way. Okay, Fleming can't see anything in a Fleming in a clear way. But when this more powerful or the compound microscope come, okay, so now the scientists means they start doing discovery in a more thorough way. Okay, and now since they are having the special techniques and more powerful microscope. Okay, so now he again they again saw in that powerful microscope, okay, some other pharaoh. And then when he saw, so they call it chromosomes, and they saw their means they saw the chromosomes and their parts as well. And through this they means they are able to know more about chromosomes. Okay, because now the microscopes are compound microscope. Okay, so in that compound microscope you can visualize the thing more deeply. Okay, that's why now the study of chromosomes become more easy. Okay, due to that compound microscope. So what is this salamander that I told you? This organism like lizard. So Fleming firstly see that the division of cells, the division of chromosomes in this particular organism only. Okay, salamander. So this is salamander, in which the Walter Fleming see the change. Okay, and see the dividing rapidly division of larva of this salamander. Okay, now let's see what a chromatin means. How chromatin the material of chromatin made up of. Okay, so chromatin material are consist of two fiber. Consist of the fibers forming by two substances. The first one is DNA, which is also known as deoxyribonucleus. Okay, deoxyribonucleic acid, which is about 40%, and the histone, which is a protein, it is of about 60%. These both things form the chromatin. Okay, these both things form the chromatin only. The DNA, that is sorry, deoxyribonucleic acid, which is about 40% inside the chromatin, and the histone, which is about 60%. Histone is a type of protein, histone protein. Okay, which wrap the chromosomes. Okay, so Molecular structure of DNA. A single DNA molecule is very large and hence it is described as a macromolecule. It is composed of two complementary strand bond around each other in a double helix. ओके सो जो मॉलिकुलर स्ट्रक्चर होता है डीएनए का, सिंगल जो डीएनए का स्ट्रक्चर होता है बहुत बड़ा होता है और वो और इसलिए मैक्रोमोलिक्यूल होता है, उसे मैक्रोमोलिक्यूल कहा गया है क्योंकि वो किससे बना होता है, डबल हेलिक्स से बना होता है. ओके लाइक दिस यू कैन सी डबल हेलिक्स यहाँ पे. ओके and next is each single DNA strand is composed of repeating nucleotides which are made up of three components: phosphate, sugar arranged lengthwise, and the nitrogenous base attached to the sugar involved to the join the complementary nitrogenous base from each other strand. Okay. Now, thus the two strands together make a ladder-like arrangement. Four types of nitrogenous base are found in the DNA. We have four types of nitrogenous base in the DNA. Okay. The first one is adenine, then guanine, then cytosine, and thymine. Now, this adenine, okay, this adenine and this thymine, these both pair with a single hydrogen bond. Okay, means A single hydrogen bond and T like this, and guanine and cytosine, these both pairs with the three hydrogen bond like this. A single bond like this, we represent the single bond, then T, and next is guanine triple bond cytosine. Yep, this is cytosine. Yeah, like this we represent triple bond. Okay, like this, like this. Okay, and single bond like this. Okay, so like this they pair each other, adenine by thymine and guanine by cytosine. Okay, adenine by thymine with single hydrogen bond, and the guanine by cytosine by triple hydrogen bond. Okay. Now this protein, histone protein, what is this histone protein? As I told you that it is a type of protein that package the DNA. Okay, that coil and packaging of the DNA into a structural unit that are known as the nucleosome. Okay, see, this protein is known as the histone protein. These yellow balls like structure is known as the histone protein only. Okay, and when they bind this DNA, when this DNA bind this means 8 histone protein, then this become nucleosome. Okay, then this become nucleosome. Next, from this chromosome this DNA go with that DNA coil the histone protein. Okay, and the pair of this histone proteins are known as nucleosome. That's it. This is the thing. Okay, so what is nucleosome? Nucleosome nothing but the 8 pairs of histone proteins which is coiled by the this DNA. Okay, this is nucleosome, and the DNA strand binds around a core of 8 histone protein known as histone octamer. Since this DNA bind the 8 histone protein, okay, 4 upper side, 4 at lower side, so 8 total, 8 histone proteins. That's why they are known as the octamer. Octa means 8. Okay, octamer. Okay, histone octamer, that's why it is known as. Now next is structure of chromosomes. What is the structure of chromosomes? You can see here, this is the daughter chromosomes. Okay, these are the daughter chromosomes. Okay, this is known as the P arm of the chromosome. Okay, it is known as the P arm of the chromosome, which is the short arm. You can see this is short arm, is it? And this is long arm. Okay, and this is the centromere, which divides the two sister chromatins. Okay, This is the centromere which divides the two sister chromatids, and this is the Q arm, which is also known as the lengthy arm or the long arm. Okay, and you can see this is the DNA molecule. Okay, this is the strands of DNA. Okay, it is the strands of DNA that form into compact structure inside the chromosomes by the histone protein. Okay, as I told you, like here it is helix, here it is DNA. Okay, so this is the structure of chromosomes. We have P arm, which is a short arm, and then we have Q arm, which is the long arm. We have centromere, which divides the two sister chromatids, and then we are having the DNA molecules. Simple. This is the structure of chromosomes only. Okay, we have that much thing inside the nucleus, inside the chromosomes. Okay, means inside the nucleus this structure exists, and not even one structure. Means uncountable, I can say uncountable chromosomes are there inside the nucleosome, inside the nucleus. Okay, you can see how much they are. Means they are very short and very much means very small, they are very tiny. Okay, so this is the structure of chromosomes. I hope this is also clear to you. Now gene, that comes to the gene. What is the gene? Okay, gene is nothing but the heredity of an organism. Okay, gene is the basic unit of heredity in a living organism. It is the gene. Okay, and gene holds the information to build and maintain cells and pass genetic traits to offspring. Means this is only the thing, this gene is the thing which means which passes the character of parents to the offspring. Okay, that's why some people say na, some people said to us that uh, uh, he is resembling to his father, and she is resembling to her mother, and his activities like his father. Okay, our grandmother has also said that he goes to his father only. Okay, and how his father did in the childhood, he is also doing the same. Okay, like this our grandmother says. So why it is so, why it is so, this is because of gene only. Okay, this is because of gene only. The next chapter, that is the third chapter, is about the gene only, the genetics. Okay, which we are going to do very rapidly. Okay, genetics. So genetics is nothing, means gene is nothing but the it is the basic unit of heredity. Okay, so this is the gene. Okay, you can see. Now next is very important and very interesting question: need for new cells. Please, why we need to a new cell? Okay, why it is mandatory to have new cells? Okay, what is the means, what is the purpose of new cells? Okay, means our old cell, our old cell, they every minute they destroy, and our not even a fraction of time, within a fraction of time our cells destroy, and they regrowth of the cell. Okay, means our red blood cell, white blood cell, these all destroy. Okay, and lakhs of uncountable of cells are making again and again, they are forming, and the old cells are destroying again and again. Okay, means I'm talking to you, means in the stock, within the stock. Okay, within just 23 minutes I lost my old cells, lakhs of old cells, I lost my lakhs of old cells, and I get my new cells too. Okay, means it is not like that only old cells lost and you will not go and get the new cells. Okay, means as soon as you lost the old cell, the new cell will occupy that particular backend space. Okay, so why do we need for new cells? The first one, for growth only. Okay, for growth only we need a new cell. Okay, means why do we say that we are growing? Because our cells are enhancing, isn't it? That's why we are growing na. Since our cells are enhancing, okay, they are enhancing in number, that's why we are growing. You can see uh, tomorrow you will be little bit, you can't see the difference, but you will be little bit means bigger than yesterday. You will be little bit, not like in a minute way. You cannot see, you cannot feel, you cannot even see that, yeah, I'm tall than yesterday. It is not like that. But it is true that from yesterday, okay, you will be little bit, not more, little bit taller than yesterday. Why it is so? Because your cells are growing day by day. It is enhancing in number day by day. And that's why now the technology, okay, now the doctors can find your accurate age. Okay, why they can able to find your accurate age means, if you are not if you not have an idea of your age, means what is your age, if you're not having an idea of it, so doctors can find your age by counting your cells. By counting your cells, doctors can find your age. Okay, so for growth is, it is also mandatory, okay, to be regrow of the cells. Okay, next is for replacement only. Replacement means how, as I told you that our old cell will be replaced by the new cells, the red blood cell, white blood cells, and the platelets. All these things will be replaced by the new one. All these old one will be replaced by the new one day by day. Okay, for replacement also it is very necessary. Next is for repair. How repair? Sorry, how repair? Suppose you got some injury, okay, within a week, it either you can see the black layer on that particular bond, or else your bond will be disappear permanently. How this is happen? Because of cell, obviously, because because of the enhancement of the cell. At that place, at that particular place, the cell occupy that vacant place, okay, where you got the bond, and that's why you will be okay. Okay, so why you apply the tubes and all these things? It just kills the bacteria, nothing else. And it helps to, means it stimulate the power of cell division. Okay, so for repairment also, okay, for reproduction, there it is also necessary for reproduction for asexual organism only. Asexual organism means for bacteria, for amoeba only. Okay, because they only divide the cell, okay, and form their offspring by division of cell. You can take an example of amoeba. This is the best example. Amoeba, what it do? What it do? It increase in length, size, and then it divides. Okay, and from the division they form two amoeba, and then they form their offspring. So like this, we need new cells. Means it has many functions, for growth also, for replacement also, for repair also, for reproduction also. As I told you, for growth. Why it is for growth? Because most organisms, including plants and animals, begin their life as a single cell, that is the zygote or the fertilized egg. Okay, this cell divides repeatedly to form a cluster of cells. Okay, for growth. For replacement, there is always a wear and tear of cells during the normal body functions. For example, 2 million red blood cells in our body are destroyed every second. As I told you, in every second 2 millions of red blood cells are destroyed, and new are forming, new blood cells are forming. Okay, for replacement means they are occupying the old one's place. Okay, next one for repair. Apart, this is apart, okay, not p part. Apart from normal wear and tear of tissues in the body, there may be accidental injuries, as I told you. Okay, if you got some bond, or if you got some injury, so you may get some cut on your skin, or fracture, or anything like this. Okay, so how you bone means again attached to that, to it, means how you bone again fixed to that original place. Okay, so this is because of the enhancement of the cell. Okay, for reproduction, as I told you, right now for asexual only. Okay, the new cells are required, like amoeba, like bacteria, for them. Okay, so this is need for a new cells. Okay, I hope this is also clear to you. Now next topic is, yep, let's see kinds of cells and the chromosomes. Suppose as I told you, in we have 23 pairs of chromosomes. Okay, total 46 chromosomes. Okay, and 23 pairs of chromosomes. So in male, obviously there will be 46 chromosomes. Is it means we have 23 chromosomes and 46. 23 pairs of chromosomes and 46 total chromosomes in man also and in female also. Okay, in male and female, okay, we have same number of chromosomes. In man we have 23 pairs of chromosomes, and in women also we have 23 pairs of chromosomes only. Okay, means total 46. So let's say these are 2n. Okay, 2n. Now sex chromosomes, that is sperm and egg. Okay, that is 23 chromosomes. So one from each uh, each organism, means one from male and one from female. That is 23 will go, and they will again combine and they will form 46. Okay, one chromosomes come, one pair of chromosomes come from male and one pair of chromosomes come from a female. That is 23 will come from each both of them. Okay, and they will form again 46 chromosomes, and that 46 chromosomes will be a new baby only. Okay, are you understood what I am saying? We have 40. Let it tell you in this way. Suppose this is man and this is female. This is male, this is female. We have 23 pairs of chromosomes. Okay, total 46. Suppose we got one pair from male and one pair from female. Okay, when they both add, again they will be 46, isn't it? Again they will be 46, and this 46 will be the new baby only. Means the male and the female, when they both form a baby only. Okay, they are giving their half of the number. Okay, and they both add and they will form the new baby. Okay, this I want to tell you. Now, very important topic, very important, mitosis, cell division. Okay, mitosis and meiosis, cell division. Let's see that only. Okay, then we'll see something else. Okay, so what is mitosis, cell division? Mitosis is how somatic or or non-reproductive cells division. Okay, they are somatic. Somatic means they are body. Somatic refers to body. They occur in body. The division occur in body, not in cells only. Okay. So what is mitosis? It is the body cell, isn't it? This is the somatic, means the body only. Okay. Body cell or non-reproductive cells. They are not reproductive cell. Reproductive cells are meiosis. Okay, meiosis are the reproductive cell, not mitosis. Mitosis are the non-reproductive cell. Okay, it make up most of your body's tissue and organs, like in skin, muscles, lungs, gut, hair cells. This will be formed by mitosis. Okay, these all things. And reproductive cells like eggs, and these all are the somatic cell which comes in meiosis. So in mitosis, it is the important thing that the number of chromosomes will be same. Okay, means the daughter cells each have the same chromosomes. Okay, and DNA as well. Okay, as the parent cell, means as the parent having the chromosomes and DNA, like this the offspring also have the same DNA and same this chromosome. Okay, the daughter cells from mitosis are called diploid cells. They are known as the diploid. Diploid means 2n cell. Okay, diploid cell have two complete sets of chromosomes. 2n means the two chromosomes. Okay, we have the two similar chromosomes, two set of chromosomes, like this, two set of chromosomes. As you can see, these are the two set of chromosomes. Okay, so since the daughter cells have exact copies of their parents, cells, DNA. Okay, so no genetic diversity is created through mitosis is not in normally health cell. Means as the parents' behavior are there, it will same in the children also in mitosis. Okay, so there will be no change. Okay, just the behavior of parents will go to the offspring only. Okay, in mitosis, since this is the body cell, this is occur in the body cell now, and this provide the growth and repair and replacement also for the body. Okay, so this is mitosis. I hope this is clear to you. Okay, if I ask the number of chromosomes, so how much we say that it is it is having the full set of chromosomes. Okay, and this is the diploid means 2n number of chromosomes. Okay, and if I told you that the dot the offspring and the parents are identical, so yes, the parents and offsprings are identical in mitosis. You can see here, one from female, one from male, they both sorry, this is forming the chromosomes. Okay, DNA replication, then these both are means they both are attracting by the ester is a point. Let it talk to you like this. Let it talk to you this next. Firstly, let's see the meiosis. Okay, then we'll see this also. Okay, then due to attracting folds, the both the sister chromatins will be detached from each other, and it will from here the depth will be formed. Okay, the dig is forming, so this dig will cut the two cells, and they will form the two sister chromatins, two diploid cells. Okay, so this is the function. Then we'll talk to you right now. Okay, so this is mitosis. Let's see the what is meiosis cell. Meiosis, this occurs in reproductive cell. Reproductive means the sperm and that is the female one, that is the sperm and this egg. Okay, so meiosis is the other main way cell division. Meiosis is cell division that creates sex cells. Okay, means we come to know that that the baby is a male or whether it is a male or female by this meiosis only. Okay, this determine that a baby is either a baby will be male or it will be female, means it will be boy or it will be girl. So this will be recognized by this only. Okay, because it creates the sex cell like female egg cells or male sperm cells. Okay, what is important to remember about meiosis, that in meiosis each new cells contains a unique set of genetic information. Means each set, suppose this is a set, so this is also having the genetic information, this set also having the genetic, means each set in meiosis, each set have the um, information of the genetic. Okay, now after meiosis the sperm and egg cells are joined to create a new organism, means to create a zygote. Okay, means to create a baby, obviously. Okay, in meiosis, so meiosis is why we have genetic diversity in all sexually reproductive organisms. Okay, during meiosis a small portion of each chromosomes breaks up and reattaches to another chromosomes. This process is called crossing over. Okay, this process is known as the crossing over, which process means the cutting of one chromosomes and attaching to the other one. Let me tell you if it is there. No, it is not here. Let me tell you what will happen in meiosis, children. Suppose this is a chromosome. I'm making just for information only. Sorry, let me change the color. No, let I do this. and when I do like this, okay. Okay, so what is happening? This one, this purple sister chromatin is coming to this pink sister chromatin. It is okay. It is coming to here. So the next will form like this. The next, yeah, the next will form like this. Like this means the half part of this pink one will attach to this purple one, okay. Like this, the half part of pink one will come is come from here, okay. The half part of this pink chromosome will come from come on this purple one, and this pink one will detach from this this half pink will detach from this pink one, and then it will attach to this purple one, okay. Are you got it? And this purple one, this pink one, what will happen with this pink one? This will have the half purple one, okay. This will have the half purple, means can you understand the process what is happening? Let me tell you in Hindi. ये जो पिंक वाला क्रोमोसोम सिस्टर क्रोमैटिन थी, तो क्या हुआ जब पर्पल वाले से इन्होंने अटैचमेंट की, तो जो हाफ जो पिंक वाला है वो पर्पल वाले में चला जाएगा, और जो हाफ पर्पल वाला था जो टूट जाएगा पर्पल वाले में से वो पिंक वाले में चला जाएगा। मीन्स दोनों में क्या हो जाएगा, एक्सचेंज हो जाएंगे दोनों के जो क्रोमैटिन्स हैं, दोनों के जो पार्ट्स हैं वो एक्सचेंज हो जाएंगे, ओके। जो मैटरनल होगा वो पैटर्नल पर चला जाएगा, और जो पैटर्नल होगा वो मैटरनल पर, ओके। द मैटरनल एंड पैटर्नल क्रोमोसोम्स वो दोनों मिक्स हो जाएंगे। क्यों हो जाएंगे? क्योंकि जो डिवीज़न है, जो रिडक्शन डिवीज़न है वो सेपरेट हो जाएगा कहाँ से? होमोलोगस पेयर से। होमोलोगस तो पता है ना, होमोलोगस मीन्स द सेम वन, ओके। सो यही मैं बताना चाह रहा था, यही होता है मियोसिस में, ओके। एंड इसी को हम कहते हैं जेनेटिक रिकॉम्बिनेशन, और आप कह सकते हैं इसे क्रॉसिंग ओवर एज वेल, okay. Now this crossing over is the reason full sibling made from egg and sperm cells from the same two parents can look very different from one another. और यही रीज़न होता है कि दोनों जो चिल्ड्रन, दो चिल्ड्रन हैं, वो एक ही पेरेंट्स से होकर भी दोनों चिल्ड्रन अलग अलग दिखते हैं। क्यों दिखते हैं? क्योंकि उनमें क्रॉसिंग ओवर हो जाता है, क्रॉस हो जाते हैं उनमें, ओके। सो दैट्स दिस इज़ योर मियोसिस। मियोसिस में बस आपको यही याद रखना है कि मियोसिस जो होता है वो क्या होता है, उसमें जो क्रोमोसोम्स होते हैं उनके नंबर हाफ हो जाते हैं, ठीक है, और उसमें जीन्स की मिक्सिंग हो जाती है, ओके। और जो मैटरनल और पैटर्नल क्रोमोसोम्स होते हैं वो भी मिक्स हो जाते हैं। बस यही याद रखना है आपने, ज़्यादा याद नहीं रखना है मियोसिस में। मियोसिस ज़्यादा पूछा भी नहीं जाता है, माइटोसिस बहुत पूछा जाता है। सो मेक श्योर माइटोसिस आप याद कर लें। माइटोसिस इज़ वेरी क्रूशियल, इट इज़ वेरी मैंडेटरी। जैसे कि मैंने अभी बताया कि माइटोसिस क्या होता है, ये बॉडी सेल में होता है। कहाँ आकर होता है? बॉडी सेल में, बॉडी सेल सोमैटिक सेल में, ओके। और ये क्या करता है? ग्रोथ, रिपेयर, रिप्लेसमेंट के लिए दिया जाता है हमें, ओके। एंड ये जो होता है इसमें कितने होते हैं? 2n मीन्स डिप्लॉयड सेल्स होते हैं, डिप्लॉयड नंबर ऑफ क्रोमोसोम्स होते हैं, ओके। मीन्स फुल सेट ऑफ क्रोमोसोम्स होता है। सो ये होता है माइटोसिस एंड दिस इज मियोसिस। आई होप दिस इज क्लियर टू यू। नाउ लेट्स कम्स टू दिस फेज। दिस इज़ वेरी इंपॉर्टेंट एंड वेरी मच इंटरेस्टिंग एज वेल। नाउ जो भी मैंने नाउ ये बहुत इंपॉर्टेंट है, बहुत क्रूशियल है, ठीक है, तो इससे अच्छे से समझेंगे हम। सो हम इसमें अच्छे से समझेंगे, हम समझने जा रहे हैं। सो हम सेल साइकिल को देखने जा रहे हैं, सेल साइकिल कि कैसे डिवाइड होता है, कैसे ग्रो होता है, फिर कैसे रीडिवाइड होता है। जो सेल होता है वो अपना वर्क एक ऑर्गेनाइज वे में करता है, एक मीन्स प्रेडिक्टेबल वे में करता है, और इसी ऑर्गेनाइज और प्रेडिक्टेबल वे को, इसी सीरीज़ को हम क्या कहते हैं, सेल साइकिल कहते हैं। ठीक है, अब हम इसके फेजेस देखने जा रहे हैं, इसके कैसे इसके क्या क्या फेजेस होते हैं। आज बहुत क्राउड है लिटरली, ओके, इतने दिनों बाद मैं आया हूँ वीडियो बनाने, देन ऑल्सो द क्राउड इज़ कमिंग, ओके। सो हम इसके फेजेस को समझने जा रहे हैं। इसके 2 फेजेस होते हैं चिल्ड्रन। द फर्स्ट वन इज़ द प्रिपरेटरी फेज, दैट इज़ इंटरफेज, ओके। एक नॉन डिवाइडिंग फेज होता है और प्रिपरेटरी फेज कह सकते हैं इसे इंटरफेज, इसमें कोई डिवीज़न नहीं होता है, अभी हम वो देखेंगे। और दूसरे सारे डिवाइडिंग फेज होते हैं जिसे हम एम फेज कहते हैं, एम फेज या माइटोसिस फेज, ओके। सो हम देखेंगे एक करके हर फेज को, ओके। सबसे पहला हमारा आएगा इंटरफेज जो कि प्रिपरेटरी फेज। इसमें कोई भी चेंज नहीं होता है, इसमें मीन्स कोई भी चेंज नहीं आता है, मीन्स एज इट इज़ रहता है। क्यों? क्योंकि जो हमारा इसमें बस क्या होता है, बस जो हमारा डीएनए होता है उसकी सिंथेसिस होती है, ओके। और भी कोई चेंज नहीं आता है क्रोमोसोम्स में। ये क्रोमैटिन फाइबर्स हैं, यू कैन सी दीज आर द क्रोमैटिन फाइबर्स। और ये जो पॉइंट आपको पर्पल कलर का दिख रहा है, ये यहाँ पे ज़्यादा क्लियर होगा, ये आपका है न्यूक्लियोलस, सॉरी, ओके। एंड दीज आर द क्रोमैटिन फाइबर्स, ओके। सो अभी इन क्रोमैटिन फाइबर्स में कोई चेंज नहीं होगा। ये हमारा प्रिपरेटरी फेज या आप स्लीपिंग फेज भी कह सकते हैं, इसमें कोई भी चेंज नहीं होते हैं, ओके। तो ये तो हमारा होता है इंटरफेज। नाउ लेट्स सी व्हाट आर माइटोसिस, मीन्स व्हाट आर द अदर फेज, ओके। सो हमारा फर्स्ट फेज होता है हमारा प्रोफेज। प्रोफेज में क्या होता है, हमारा जो सेल होता है उसमें मीन्स जो हमारे क्रोमैटिन फाइबर्स होते हैं वो अब हमारे क्रोमोसोम्स में चेंज हो चुके हैं। क्रोमोसोम्स में कैसे चेंज हो चुके हैं? दे बिकम डिस्टिंक्ट एंड शॉर्ट, ओके, दे बिकम शॉर्ट एंड थिक। नाउ द क्रोमैटिन फाइबर्स बिकम क्रोमोसोम्स इन प्रोफेज, ओके। सो और जो हमारे न्यूक्लियस होता है वो भी अपीयर होने लगता, इट इज़ ऑल्सो डिसअपीयर, ओके। द जो हमारा दैट इज़ न्यूक्लियस होता है, ओके, दैट इज़ ऑल्सो डिसअपीयर, ओके, इन प्रोफेज। ठीक है, प्रोफेज में क्या होता है, हमारे जो क्रोमैटिन फाइबर्स हैं वो क्रोमोसोम्स में चेंज होने लगते हैं। सेंट्रियोल्स जो होता है, यू कैन से दिस इज द सेंट्रियोल्स, दिस वन। सेंट्रियोल्स हमारे अपोजिट पोल्स में जाने लगते हैं। अपोजिट पोल्स मीन्स ये वाला जो सेंट्रियोल्स है वो इधर आएगा, ये वाला सेंट्रियोल्स वो इधर आएगा, या ऊपर होंगे तो इधर आएगा या इधर आएगा नीचे, ओके। अपोजिट पोल्स में जो हमारे सेंट्रियोल्स होते हैं वो जाने लगते हैं। और जो सिस्टर क्रोमैटिन्स होता है, ओके, सिस्टर क्रोमैटिन्स वो भी अटैच हो जाता है सेंट्रोमेयर से। यू कैन, एज आई टोल्ड यू, व्हाट आर सेंट्रोमेयर, सेंट्रोमेयर ये होता है, दिस इज सेंट्रोमेयर, ओके। मैंने स्ट्रक्चर ऑफ क्रोमोसोम्स तो बता दिया है ना, ये सारा सिस्टर क्रोमैटिन होती हैं, ये सेंट्रियोल होता है, ये पी आर्म शॉर्ट आर्म होती है, क्यू आर्म लॉन्ग आर्म होती है, ओके, डीएनए मॉलिक्यूल होता है, ओके। ये सब तो मैंने बता दिया आपको। नाउ लेट्स सी व्हाट आर दिस प्रिपरेटरी फेज। सो प्रोफेज में कुछ नहीं होता है, बस जो हमारे क्रोमैटिन फाइबर्स होते हैं वो क्रोमोसोम्स में चेंज होते हैं, और जो एस्टर होता है वो दोनों मतलब पोल्स में जाने लगता है, पोल्स पे चला जाता है, ओके। स्पंडल फाइबर होता है वो डॉटर सेल्स में अपीयर होने लगता है, ओके, डॉटर सेल्स के बिटवीन में अपीयर होने लगता है, ओके। और जो न्यूक्लियर मेम्ब्रेन होता है और न्यूक्लियोलस होता है वो भी डिसअपीयर हो जाता है, किसमें हम प्रोफेज की बात कर रहे हैं, ठीक है। नेक्स्ट इज मेटाफेज। मेटाफेज में कुछ नहीं होता है, जो हमारे क्रोमोसोम्स थे वो हमारे एक इक्वेटर वे में अरेंज हो जाते हैं, इक्वेटोरियल वे में अरेंज हो जाते हैं मेटाफेज में, ओके। और मीन्स इक्वेटर, सी थोड़ा यहाँ जोग्राफी देख लो आप लोग, दिस इज़ द इक्वेटर, ओके। इक्वेटर होता है ना, तो इक्वेटर वे में अरेंज हो जाते हैं क्रोमोसोम्स मेटाफेज में, ठीक है। और सारे की सारी जितने भी क्रोमोसोम्स होते हैं ये सब स्पिंडल से, ओके, अपने अपने स्पिंडल से अटैच हो जाते हैं भाई सेंट्रोमेयर मेटाफेज में, ओके। नाउ एनाफेज में क्या होता है, जो भी क्रोमैटिन होता है, ओके, हर क्रोमोसोम्स का जो क्रोमैटिन होता है वो सेपरेट हो जाता है और पोल्स की तरफ अट्रैक्ट होने लगता है थ्रू एस्टर। ये एस्टर होते हैं, ओके। ये एस्टर जो है वो इन्हें अट्रैक्ट करने लगता है, ओके। अब यहाँ पे ये क्रोमोसोम्स में इक्वेटर वे में अरेंज हुए थे, अब ये अलग हो गए एक दूसरे से सिस्टर क्रोमैटिन। अब ये किसके थ्रू अलग हो गए? थ्रू एन अट्रैक्टिंग पुश through an attracting pull by ester, okay, in anaphase. So now let's see what happens in telophase. telophase में क्या होता है? जो हमारे क्रोमोसोम्स के डॉटर क्रोमोसोम्स होते हैं वो सब हमारे अपोजिट पोल्स में जाने लगते हैं, ओके। और एक डैप्ट बनने लगती है। डैप्ट मीन्स एक, यू कैन सी हेयर ऑल्सो, ये एक डैप्ट बननी है और इसी से हमारे दो सेल्स हमारे डिवाइड हो जाएंगे, ओके। इसमें क्या होता है? जो भी न्यूक्लियर मेम्ब्रेन होता है वो फॉर्म हो जाती है और न्यूक्लियोलाई जो होता है हमारा, न्यूक्लियोलाई जो हमारा किसमें प्रोफेज में फॉर्म हुआ था वो हमारा इसमें टीलोफेज में या टीलोफेज में हमारा डिसअपियर हो जाता है, रीअपियर हो जाता है। जो हमारा और एम सही न्यूक्लियाई होता है वो रीअपियर हो जाता है टीलोफेज में, जो हमारा प्रोफेज में एपियर हुआ था, डिसअपियर हुआ था, ओके। जब हमारा टीलोफेज की बात हुई, ओके। टीलोफेज में जितने क्रोमोसोम्स होते हैं वो पोल्स पे चले जाते हैं और ज़्यादा डिफ्यूज़ हो जाते हैं, ओके, और ज़्यादा डिफ्यूज़ हो जाते हैं। एंड द न्यूक्लियर इवॉल्व इज रिफॉर्मिंग द साइटोप्लाज्म में बी डिवाइडिंग, ओके। एंड नेक्स्ट हमारा होता है साइटोकाइनोसिस। साइटोकाइनोसिस मीन्स साइटोप्लाज्म का डिवीज़न, ओके। मीन्स जो हमारा साइटोप्लाज्म होता है वो फाइनली दो डॉटर सेल्स में चेंज हो जाता है। जो हमारा ये डैप्ट बनना था, यहाँ पे गड्ढा बनना था, है ना, ये डिग बननी थी यहाँ पे, यही डिग अलग कर देती है इन दो सिस्टर क्रोमैटिन को, यही अलग कर देती है दो सेल्स को, ओके, डॉटर सेल्स को। तो ये तो हमारा था सब, यही सेम एनिमल सेल में होता है, ठीक है। रेस्टिंग फेज आती है जिसमें कोई भी डीएनए का अभी कोई भी सिंथेसिस नहीं होता है, ओके, रेस्टिंग फेज में। देन प्रोफेज आती है जिसमें हमारे क्रोमैटिन फाइबर्स क्रोमोसोम्स में चेंज हो जाते हैं और जो हमारा एस्टर होता है वो अपोजिट पोल्स में जाने लगता है। देन मेटाफेज में हमारे इक्वेटर वे में खड़े हो जाते हैं, जितने भी लग जाते हैं इक्वेटर वे में, और एस्टर के थ्रू वो अट्रैक्ट होने लगते हैं। क्योंकि एस्टर के थ्रू अट्रैक्ट होने लगते हैं तो उनसे जो अट्रैक्शन होता है वो बहुत बढ़ जाता है, जिससे हमारी जो सिस्टर क्रोमैटिन होती हैं वो अलग अलग हो जाती हैं और वो सब पहुंच जाती हैं अपनी अलग अलग वे में। मीन्स किस में पहुंच जाती हैं? एस्टर की तरफ पहुंच जाते हैं, जिससे हमारा टीलोफेज में क्या हो जाता है? हमारा सेल डिवाइड होना शुरू कर देता है एंड अगेन जो भी न्यूक्लियाई था वो रीअपियर होने लगता है टीलोफेज में, ओके। स्पिंडल फाइबर भी डिसअपियर हो जाता है और साइटोकाइनोसिस मीन्स साइटोप्लाज्म के डिवीजन में क्या होता है? दोनों डॉटर सेल्स फॉर्म हो जाती हैं, ओके। सो ये था हमारा डिवीजन ऑफ सेल एंड फॉर्मेशन ऑफ द chromosomes. Now let's see what is the significance of mitosis. Okay, significance of mitosis. So mitosis is important for sexual reproduction indirectly. Okay, mitosis किसके लिए important होता है? Sexual reproduction के लिए. It allows the sexually reproducing organisms to grow and develop from a single cell into a sexually mature individual. ये allow करता है एसेक्सुअल रिप्रोडक्शन होने पे। मीन्स जो हमारे अमीबा होते हैं, जो हमारा बैक्टीरिया होता है, वो सब हमारे एसेक्सुअल रिप्रोडक्शन से रिप्रोडक्शन करते हैं, एसेक्सुअल मोड से। सो ये कैसे पॉसिबल है कि एसेक्सुअल मोड से रिप्रोडक्शन होता है? ये इसी के थ्रू होता है, माइटोसिस के थ्रू। यही अलाउ करता है एसेक्सुअली एनिमल्स में कि एसेक्सुअल मोड से अपना रिप्रोडक्शन करके अपने इंडिविजुअल को एनहेंस करिए, ओके। नेक्स्ट है दिस अलाउस ऑर्गेनिज्म टू कंटिन्यू द रिप्रोड्यूस थ्रू द जनरेशंस। जैसे मैंने अभी बताया कि ये जनरेशन के लिए बढ़ाते हैं अपने ऑफस्प्रिंग्स को बाय एसेक्सुअल रिप्रोडक्शन। ये तो सिग्निफिकेंस है माइटोसिस का, ओके। माइटोसिस के सिग्निफिकेंस में भाई यही याद रखना कि हमारा जो माइटोसिस होता है वो सेक्सुअल रिप्रोडक्शन इम्पॉर्टेंट होता है। ओके, किसके लिए? सेक्शुअल रिप्रोडक्शन के लिए। ये अलाउ करता है सेक्शुअली रिप्रोड्यूसिंग ऑर्गेनिज्म टू ग्रो एंड डेवलप फ्रॉम अ सिंगल सेल इनटू अ सेक्शुअली मैच्योर इंडिविजुअल। ओके, and this allow organisms to continue to reproduce through the generations. Okay, now next is meiosis का significance. meiosis का significance क्या कहता है? It brings about reduction. ये reduction लाता है chromosomes के numbers में, means haploid से only diploid से only haploid condition में रह जाता है, 2n से only n में, means reduction हो गया, 2n से n हो गया तो reduction हो गया। ओके, क्योंकि इसमें रिडक्शन हो रहा है तो क्रोमोसोम्स के नंबर को मेंटेन रखना भी बहुत ज़्यादा इम्पॉर्टेंट है। ठीक है, और ये प्रोवाइड करता है चांस फॉर द अपीयरेंस ऑफ न्यू जीन कॉम्बिनेशन एज अ रिजल्ट ऑफ क्रॉसिंग ओवर। ओके, और इसी यही सिचुएशन क्या करती है? वेरिएशंस ला देती है। ओके, एंड नेक्स्ट इज नेसेसरी फॉर द फॉर्मेशन ऑफ गैमीट्स इन एनिमल्स एंड स्पोर्स इन प्लांट्स। ओके, मीन्स जो एनिमल्स हैं उसमें गैमीट्स का फॉर्मेशन करती है, और जो प्लांट्स है उसमें स्पोर्स का। ओके, ये हमारा सिग्निफिकेंस हो गया मियोसिस का। ओके, नाउ लेट्स सी द डिफरेंस बिटवीन माइटोसिस एंड मियोसिस। अभी तक हमने बहुत ज़्यादा लेंदी वे में दिखा था माइटोसिस और मियोसिस। ओके, अगर फिर भी आपको समझ नहीं आया है तो हम इधर देख लेते हैं माइटोसिस और मियोसिस। ओके, and if you want to write in your examination you can write these differences also. In mitosis occur in all organisms except viruses. Okay, सिर्फ viruses bacteria इनको छोड़ के सारे organisms में occur होता है। Okay, इसमें वही meiosis होता है वो plants में, animals में, fungi में, इन सब में occur होता है। ठीक है, माइटोसिस किस में अकर होता है? वायरसेस को छोड़ के हर ऑर्गेनिज्म में अकर होता है। वही मियोसिस किस में अकर होता है? सिर्फ एनिमल्स में होता है, प्लांट्स में होता है, फंजाई में होता है। एनिमल्स मतलब हम भी आ जाते हैं एनिमल्स में। ओके, ह्यूमंस विल आल्सो बी कंसिडर इन द एनिमल्स। ओके, माइटोसिस में नेक्स्ट पॉइंट क्या है? क्रिएट्स बॉडी सेल्स। क्या करता है? बॉडी सेल्स को क्रिएट करता है। वहीं हमारे मियोसिस में सेक्स सेल क्रिएट्स होते हैं। ठीक है, माइटोसिस में ये याद रखना है हमारे सोमैटिक सेल्स क्रिएट्स होते हैं। सोमैटिक सेल्स मीन्स बॉडी सेल्स, और मियोसिस में हमारे सेक्स सेल्स क्रिएट्स होते हैं। ओके, माइटोसिस का थर्ड पॉइंट क्या है? वन सेल डिवीज़न, ओनली वन सेल डिवीज़न होता है। भाई मियोसिस में टू सेल डिवीज़न होता है। ठीक है, माइटोसिस में नेक्स्ट पॉइंट क्या है? नो रिकॉम्बाइनेशन, कोई भी कॉम्बाइनेशन नहीं होता है। वहीं मियोसिस में क्रॉसिंग ओवर या कॉम्बाइनेशन हो जाता है। ओके, नेक्स्ट माइटोसिस में क्या होता है? प्रोड्यूस टू डिप्लॉइड सेल। ओके, प्रोड्यूस टू डिप्लॉइड डॉटर सेल्स। वहीं मियोसिस में 4 हैप्लॉइड डॉटर सेल्स प्रोड्यूस करते हैं। ओके, मियोसिस में टू डिप्लॉइड सेल्स। ओके, वही माइटोसिस में 2 डॉटर डिप्लॉइड सेल्स होता है, वही मियोसिस में 4 डॉटर डिप्लॉइड सेल्स को प्रोड्यूस करते हैं। ओके, 4 हैप्लॉइड नॉट डिप्लॉइड। डिप्लॉइड का मीनिंग होता है 2n, हैप्लॉइड का मीनिंग होता है ओनली n मीन्स सिंगल। ठीक है, एंड माइटोसिस में लास्ट पॉइंट है डॉटर सेल्स आर आइडेंटिकल। डॉटर सेल्स आइडेंटिकल होते हैं मीन्स जैसे पेरेंट्स होंगे वैसे ही ऑफस्प्रिंग होगा। वही मियोसिस में ऐसा कुछ नहीं होता है। ओके, डिफरेंट हो सकते हैं। Okay, so creates new cells, follows same steps, begin with one parent cells. So this is the difference between mitosis and meiosis. Okay, so this is, now let's come to the homework question. Okay, last part of this session, homework question. So yep, fill in the back end position. First question is saying chromosomes are highly coiled and condensed क्रोमैटिन फाइबर्स। ओके, क्रोमोसोम्स हाईली कंडेंस और क्वाइल्ड फॉर्म किसके होते हैं? क्रोमैटिन फाइबर्स के। क्रोमैटिन फाइबर्स कंडेंस्ड एंड फॉर्म क्रोमोसोम्स। दोनों अपोजिट क्वेश्चन दिए हैं, इजंट इट? क्रोमैटिन फाइबर्स कंडेंस्ड एंड फॉर्म क्रोमोसोम्स। इंटरफेज इज ऑल्सो नोन एज what? It is known as the preparatory phase, resting phase. Okay, not preparatory phase, resting phase. Interphase is also known as resting phase because उसमें अभी कोई डीएनए का चेंज नहीं आता है, डीएनए एज इट इज़ रहता है। ओके, अभी कोई भी चेंज किसी में भी नहीं होता है, वो रेस्टिंग फेज होता है। ओके, नाउ नेक्स्ट इज एग्री एंड डिसएग्री। agree and disagree. The center part which attaches the two sister chromatids is called centromere. Obviously, agree. Okay, it is true. Next is the specific sequence of nucleotides on a chromosome constitutes a chromosome centromere. It is false. Okay, it is the central part which attaches the two sister chromatids is called the central part which attaches the two sister chromosomes is called the centromere. Yeah, true. First one is true. Second one, okay. Second one, the specific sequence of nucleotide on a chromosome constitute a chromosomes. False. It is false. So the specific sequence of nucleotides on a chromosome constitute a chromosomes. Disagree. It's constitute a genes. Okay. What is constitute a gene? Okay. So this is agree and disagree. Now, who am I? Who am I? I am the resting phase in the karyokinesis. Who am I? What is the resting phase? Obviously, we right now I discuss the interphase. Okay, interphase is the resting phase. Now next is I am the protein which help in the coiling and packaging of DNA into structural unit called nucleosome. Can you recognize me? Yeah, you are histone protein. Okay, so we all have done this homework question. Okay, so first one will be the chromatin fibers, next one will be the chromosomes, third one will be the interphase, is also known as the resting phase. Okay, now agree and disagree. The central part of the central part which attaches the two sister chromatids is called a centromere. True. The specific sequence of nucleotides on a chromosomes constitute a chromosomes. False. It constitutes a gene. Okay, the right answer is gene. It constitutes a gene. Here it will come gene. Now, who am I? I am the resting phase in the karyokinesis. Who am I? You are the that one, the prepare phase, or the prepare phase, that is known as the resting phase, interphase. Okay. Next is I am the protein which help in the coiling and packaging of DNA into structural and structural units called nucleosome. Can you recognize me? Yeah, you are histone protein. So I hope this chapter chromosome is clear to you, and this is very important. Mitosis and meiosis difference. Okay, mitosis and meiosis difference. Mitosis and meiosis difference is very important. The sound was so horrible, right? So yeah, I hope this chapter is clear. Homework question is also clear to you. So yeah, bye bye, and yeah, if you love this video, so don't forget to give this video a big thumbs up, subscribe my channel if you didn't do it yet, and press the bell icon. Okay, this chapter was so lengthy. I hope this is clear to you. The structure of chromosomes, cell cycle, and cell division. Okay, we had we have seen all the things in a very good manner. Okay, if you have any doubt, please tell me in the comment section, and just read this part, significance of mitosis and significance of meiosis and difference between mitosis and meiosis once again, and this one as well. Don't forget to read this one. Means no need to read this mitosis and meiosis so lengthy. Just read this one, mitosis and meiosis. That's why I applied this image, so that it will be easy to you. Okay, so yeah, that's it, and bye bye. Stay tuned for more videos. So yeah, bye, have a good day.